Welcome to our webinar series for professionals.
Welcome everyone for the February webinar on biological aging test or markers to find out if we are the age we are, or if we are younger or older, or stuff that we can do in order to support longevity and support the long healthy life for sure. I'm Anne-Catherine, co-founder of Nordic Laboratories and DNA Life, where we are focused on functional medicine and personalized medicine and poking and provoking in regards to creating optimal health in people's lives and creating awarenesses and what information can we gain and how can we use this information as a tool in order to both motivate us in creating healthier lifestyle choices and living the healthiest life as possible. So today with me I have Hannah Wendt. She has a degree in biology, and she's the director of operations from TruDiagnostic laboratory, where it's taken her and I over a year to actually get this webinar on feet, where we agreed, like in September 22 it was, let's get going, and I did my test, and I was just very excited. But things takes their turns, and but we've managed to get the test in the system now. So the tests that Hannah will be presenting and talking about today are already available in VMS, so you can go in and order them. They have just been released actually today. So when you get off the hook here, and if you feel eager for shopping, then the products are available for you for sure. So and then having said that, before I just pass on the stage to Hannah, we are of course recording as always, and we will also be sharing Hannah's slides, so that you will get that in an email, both the recording and the slides, sometime probably next week. And then I just want to remind you that we do have a live education conference coming up, and that's very soon. It's the first weekend of March in Dubai. And of course, we continue with these webinars and to share news and further education with you all as much as possible. So having said all that, write in the chat box, ask your questions that you have along. I'll take note of them and try to convey them at the end of the Q&A, where Hannah then will help me answering the questions, and hopefully we'll have a fun time and learn lots of exciting things. And actually, just the last thing, I just want to do a little anecdote here, because during COVID, I actually did my own biological age test with a different company. I'm not gonna say the name because it seemed too simple. But I really liked the test because it just gave my age compared to my biological or chronological age. So, and I was 7 years younger then. So I was very happy and really liked that lab. But then it was done on other people, and I could see it just didn't really make sense compared to lifestyle. So I questioned the test, and it wasn't actually until I met Hannah in September 2022, where I had been looking at the TruDiagnostic for a while, and I also know that Jeff Bland was doing some research through TruDiagnostic in regards to some of the projects that he's going on, and I just felt more and more confident that TruDiagnostics was going to be our partners in regards to biological age. So just so you have an idea that I've been looking at this for several years now, and I don't just go with anyone. I kind of need to feel a little certain. So just we've done the due diligence, and hopefully you trust us when we do our due diligence, and then so that it's easier for you to partner up if you want to do biological test aging on your patients or on yourself. So enough from me. Hannah, I'm going to pass on the stage to you so you can share your screen and take over the mic. I'll unmute myself and be quiet and just sit here and look forward to your presentation and the Q&A after your presentation.
Awesome. Thank you so much for that wonderful introduction, Anne-Catherine. I really appreciate your time. It's nice to see everyone here today. I actually recognize some familiar names. So if I have met you before, welcome back. I hope you are able to listen in and learn something new. And if I haven't, as Anne-Catherine said, my name is Hannah Wendt. I'm the co-founder of TruDiagnostic, and I am the director of operations, usually working with our healthcare providers and educating about our test, what it means, really how to use these results and interpret them, and then use them in the clinic as well to drive your decision making. So I want to just review what I hope we can learn on this presentation today. And that includes the following points, which is why biological aging is important, how to actually measure the aging process, the difference between genetics and epigenetics, applications of epigenetics and DNA methylation algorithms, which we'll define, comparison of epigenetic clocks. As Anne-Catherine said, there are a lot of other companies out there and even just different clocks. We need to know really what their advantages and disadvantages are and how they compare head to head. And then I want to introduce our new Omic M-Age, which is the most predictive clock that has ever been created to date with our collaborators at Harvard. So we'll save that for the end. And there are some slides that I'll just go through really quickly that I will, include in the presentation that we send to you afterwards. But I do want to leave a little bit of time at the end for questions as well. So without further ado, I want to start with a little, trivia Family Feud style game. You see all of these diseases out here, arthritis, cancer, kidney disease, CVD, etc. Does anyone know, you can pop it in the chat if you want to play along, what the #1 risk factor for all of these conditions is? I'll show you another slide as well. These are the top 3 causes of death in 2020 for the US, your heart disease, cancer, COVID-19. I blocked the answer out for you all, but you can see some risk factors here, maybe things like, sex, right? I have chronic inflammation. Someone who actually guessed the answer right already. It's the first one in the chat, which is aging. So thank you so much. You're exactly right. Aging is that #1 risk factor for every single chronic disease and death. So we need to treat it as such. We need to treat it as a primary outcome where everything else is going to be secondary. Usually, we're stuck treating these secondary outcomes when we need to look inward and get to really that root cause. You can also see just how aging, which again is really not recognized as that risk factor, is trumping over every other one on these x-axis labels here. So the aging is super important, and we can now measure this process actually through epigenetics. So what is epigenetics? Well, a lot of people think of it like a light switch. It's really about the study of our behaviors and environment and how this can actually change the way our genes are expressed. So I like to explain this to my providers and have them explain it to patients in the same way because it's really simple to understand. Our eyes and our heart, for example, have the same genetic information, right? Genetic testing is going to be defined on this left-hand side here. But what makes our eyes and our heart? Well, it's actually epigenetics. It's how we are expressing our genes to end up with that phenotypic outwards behavior, that phenotypic outwards expression. So you can just see a little bit of a difference, these tables here comparing genetic testing versus epigenetic testing. Keeping in mind, genetic testing your genetics are never going to change, whereas your epigenetics are. Right, we're able to control that genetic expression. So I want to make this as relatable as possible to you in your everyday life. Why should you be caring about this? And there are some really cool applications for epigenetics. But I do want to preface by saying there are a lot of other epigenetic modifications out there, and in particular, we're looking at DNA methylation. Really, with the definition being if something is methylated, you're turning that gene off. Vice versa, if that methylation is not present, you're expressing that gene and turning it on. So we're going to start with the agouti mouse study, actually. You all may heard of the study or be super familiar. This is going to be by Dr. Randy Girdle. It is the first ever study really looking at epigenetics and the manipulation of specific genes called the agouti gene in this case. to really study the effects of supplementation on coat color and obesity. What you can really see is when this, mouse here at the top is given supplementary diet capsules and pills, you are going to get a healthier outcome or outwards expression. This is going to be, brown and healthy mice. This is where that agouti gene is actually silenced. It is going to be methylated, as you can see in this slide here, where with no dietary supplementation, you're going to get that unfavorable phenotype. This offspring is going to be mostly yellow and unhealthy. So this agouti gene is unmethylated and activated, as you can see here. So we are really, this was again, the first ever proof of concept study showing we can actually control and manipulate genes through expression through methylation using dietary supplemental pathways. Another cool application, I don't know if any of you are familiar with who this person is here. This is Natalia Grace. Maybe you all have seen the documentary, The Curious Case of Natalia Grace. If you haven't, that's okay. It's on HBO Max, where TruDiagnostic was featured. She actually flew to our facility so we could measure her chronological age with DNA methylation and epigenetic information. The reason being is she is an orphan. She really did not have any information on when her birth date was, and she has a form of dwarfism. So physically, it's really hard to gauge maybe where her chronological age would lie. So this is another application. So we have the agouti mouse study where we can actually change expression levels through supplementation, diet, and these chronologically trained clocks as well. The last one I want to show you is more clinically applicable. Schizophrenia only used to be diagnosed through healthcare providers. You had to physically go into the clinic. Now we can diagnose it with DNA methylation. All right, in this study here, I won't go too in depth, but if you're interested, you can take a look and I can send you the paper. So we've talked about aging being the biggest risk factor for disease and how we measure this through DNA methylation and epigenetics. But now I want to talk about the applications for this testing. Why is biological age testing the #1 biomarker you should be measuring in your clinic? So I'll go through a couple bullet points here, but first of all, you're going to hear me say this over and over again. You've already seen these images once. It is the biggest risk factor for chronic disease and death. I will sound like a broken record when I say that, but that is something we've known for a very, very long time. So we have to treat it as a primary outcome. And in order to manage it, we absolutely need to be able to measure it. Number 2, it is correlated to quality of life metrics. IQ, balance, muscle mass, grip strength, gait speed, cognitive functions. You can see some of these images here where if you have faster aging, you're going to be worse off in all of those categories. And I do not know one person who has ever wanted more poor quality of life outcomes. All right, this is a really interesting image too, where all of these people here are 45 years old chronologically, but they look very, very different. Okay, so this is even going to be present and be able to be tracked through facial aging. We know that chronological age is not a good marker for this because, we know some people who are 30 that look like they're 50 and some people who are 50 that look like they're 30. That's the need for the biological aging. Accelerated aging equals a shorter lifespan and health span, and deaccelerated aging equals a longer lifespan and health span. So it's about your quality life at the end of the day. What do you want to do at the end of your life? For me personally, I know I want to, live for a very long time, but I want to be able to still be functional and do what I was able to do when I was still in my, 40s or 60s, when I'm getting closer to 80s and even 100, hopefully. Number 5, aging starts as soon as we are born. So aging is a very exciting tool that we can use early on. I highly recommend, if you all haven't even tested yourselves, to just get tested now. Funny enough, most of the people we test at TruDiagnostic is between 40 to 60 years old chronologically. In that same time frame, your disease diagnosis skyrockets. So it's never too early to really start testing. You're never too old to really start testing chronologically, that is, but we do encourage people to do this as soon as possible so you can make changes and start the longitudinal testing, which is absolutely key. It's great to go ahead and test once, but to again affect change, we need to test continually. And we can talk about some of those retest windows and periods as well. Last one here, aging starts as soon as we are born. Oh, sorry, I already said that one. So aging optimization can be a great functional testing and preventative tool. Again, harping on that, it's a tool that we need to use as soon as possible. So why wouldn't everyone try and measure and treat their major risk factor for all of these diseases, right? Especially if you are currently healthy. So I hope you're starting to understand, the usefulness of this test, how it can be applied into your clinic, and to harp again on the repeating test time. Most users are doing this as a baseline and type of in a package program or membership model, which we won't get too much into today, but we can always talk about later. And then they're going to repeat it 6 months later, at least 12 months later, or somewhere in between, to test the measurements and see the effectiveness of treatment. So you're no longer wasting, money, resources, time guessing on therapies, which are going to target that risk factor. So with that being said, there are a lot of other biological age testing out there on the market. And again, I'm not going to go too in depth on each one, but I do want to mention how TruDiagnostic is different, because this is really, really important, that you understand you are using the best technology that is available. So here are just some examples of what's out there. Number one, the main difference is TruDiagnostic only uses peer-reviewed published algorithms to interpret the DNA methylation data that we are gathering. All right, so you can see how we compare to some other companies that use some older generation clocks, or even other more consumer-based companies that aren't going to use published algorithms. And we do say this internally a lot. It's kind of an inside joke that I'll share with you. But we say that it's like going to a fortune teller if you're using companies that don't have published algorithms, because you just have to trust that what they're telling you is true. You have to trust to believe it when there's really no proof behind it. But even though that's more of an inside joke, it does hold some truth to it, right? So again, we are really the only company that is going to use published algorithms. You can see in this column here. Secondarily, these algorithms are only going to be available, and this testing is only going to be available through a blood sample. So it is a blood spot card, still going to be an invasive sample. It's a very small blood spot card. It's like the quarter size. This is extremely important, though, because the algorithms which have been validated have only been used in blood. All right, where we're not going to settle for an easier collection method such as saliva, even though it's just a little bit easier, because it hasn't been validated quite yet. And the next difference I want to talk about is the really expansiveness of epigenetics, or the generation of these different clocks. So when you're comparing the companies head to head, TruDiagnostic is the only company that uses second and third generation clocks. And if you're not familiar with this, I'm going to give you a little bit of a history lesson behind this. And this is very, very important to understand these differences. So I'll walk you through left through right, starting with the first generation clocks. Please don't get caught up too much in this image. It is a little bit busy, but I'll walk you through it. So these first generation clocks on the left-hand side were really created in 2011, 2013, and between there, as a huge breakthrough in age research and science. So at the time, there were really many reasons this is exciting. The predictability of these clocks was amazing. We all know, again, that age is that biggest risk factor for every single chronic disease and death. And it was immediately clear that these clocks right here were better than chronological age at telling us how a patient was aging. And therefore, their risk for almost every single chronic disease was measurable. However, the first generation clocks were actually used to predict chronological age of the patient it tested. That is the definition of a first generation clock. And the problem with that is we don't care about chronological age. People like Natalia Grace that I showed you in the beginning do care, because they're put in unfortunate circumstances and situations. But we really care about the biochemistry of aging, right? That doesn't mean there still aren't applications for these. They're even used at crime scenes to see how old DNA samples are, to see if refugees are old enough to seek asylum. So they have their utility, but clinically they do not mean anything. All right, so how can we detect that actual true biological aging signal better? And the answer to that question is to measure these and train these DNA methylation patterns to better measurements of aging rather than chronological age. And this is how these second generation clocks were created right here, this green column. Now, the three most popular second generation clocks that were previously documented in the literature are going to be Dr. Morgan Levine's PhenoAge. So with DNA methylation and epigenetics, they're also going to use blood-based measurements. Grim age, which is a death predictor, they use 12 protein-based measurements, and they train this for time until death. And lastly, DNA methylation, telomere length. Still a second generation clock because you're using telomere length data on top of the DNA methylation data to predict telomere length. So these clocks were much better than first generation clocks. And how do we know that? Well, accelerated aging scores were even more predictive of negative health outcomes, and deaccelerated aging scores were more predictive of better outcomes, lower disease risk. All right, so these second generation clocks are more associated, highly associated with disease. So we have first generation, second generation. What is third generation? Third generation is going to be hopefully pretty easy for you all because there's only one third generation clock, which is this Deneen pace algorithm right here. So to get the best aging signal, we need to follow individuals across their entire life points, their entire lifetime across multiple time points. And this is exactly what the Deneen pace did. So unlike previous clocks, the Deneen pace is not trained on chronological age. It is the first clock to be trained entirely on phenotypes of aging on patients across their lifetime. This study was done in Dunedin, New Zealand, starting in 1972 and 1973, and it's still going on today. It's a fascinating study, one that I would also read the paper to, and I could talk to you forever about. It's my favorite to speak on. And this study is still going on. As I mentioned, they have a 96% retention rate, which is insane. So this is super helpful because we're really not picking up noise for measurement. So when you're following the same individual, we can make sure that things like environmental exposures aren't included in these clocks, right? So one example of that is, 50 years ago, many people were exposed to more lead, right? Through leaded gasoline, less antibiotics, less microplastics. And if we don't control for those at the time which people lived, the algorithm might include more markers associated with exposures rather than just aging. So this is a super unique clock that was created in collaboration with our investigators at Duke and Columbia, Dr. Terry Moffat, Ashlam Caspi, Daniel Belsky as well. So this is more of a speedometer, kind of a point in time, capturing a 3 to 6 month trailing average, where the first and second generation clocks are going to give you an actual kind of age. All right, so those are the generation of clocks. Hopefully you stuck with me there. Now, what else sets TruDiagnostic apart in terms of the published clocks? Well, we just created our very own new Omic M-Age with Harvard. It is a second generation clock. It is the one I mentioned at the beginning that we would cover, and it is the best biological age clock ever to date. Best is subjective, but I do have the points to prove that, and I'll walk you through that. So I want to introduce our new Omic M-Age. I'll introduce this just very briefly, but then I'm going to go back and tell you again why it's superior. So when TruDiagnostic was created back in July of 2020, we really set out on a mission to create the best scientific algorithm that has ever been created to date. And we did this with the Omic M-Age. We really succeeded here. And we partnered with researchers from the Harvard's University and Partners Biobank, which includes 50 years worth of samples that we were able to collect and look at all of the data points in between their lifetime. So this process has almost taken us about 3 years to finalize, but super proud to obviously be able to release it to you all now. This even goes over metrics that can represent increase or decrease in several diseases you can see here. And it actually tells you why you're aging as well. So this is the idea of epigenetic biomarker proxies, where we can use epigenetics to predict other omics. This is again, very, very fascinating. I'll talk on this more as we go through it, but we can use epigenetics to predict genomics, transcriptomics, proteomics, metabolomics that are, positive for your aging or maybe negative for your aging, and tell you what to work on as it relates to, again, that overall Omic M-Age. So first and foremost, I want to give you an introduction to this algorithm, a little bit more about how we created it before going into why it's the best. So aging is very complex. We just recently went from 9 hallmarks of aging to more than 14 hallmarks of aging. And I imagine that they'll add even more in the years to come. And these papers are really interesting as well if you haven't read them. So we know that it's very multifactorial. So how do we account for this as scientists? Well, we're trying to really take an approach where we're integrating multiple layers of multi-omics, where again, we can use epigenetics then to predict anything downstream of it. So you can see the multi-ome here, where, we talk about all of these different layers. So the proteins, the metabolites, you even have your phenomics, that phenotypic outcome, you have your transcriptomics, your epigenetics, et cetera. And this is really important because we know, our health outcomes, or those phenotypes that you're going to get at the end of this multi-ohm, are a result of more than just your genomics, right? It's the epigenetic layer, it's the transcriptomic layer, where, we're reading that epigenome. It's the peptides, which are the proteins in your body. That's the proteome, the metabolites that are going to be the byproducts of those proteins. All of these are going to be crucial factors in how our biology operates. So everything I just described there is again, that idea that everything is interconnected, all these cellular processes together. That's what we represent here as the multi-ohm. So you can see what we did and what we measured to create the algorithm. About 27,000 untargeted proteins, 61 clinical lab variables, 8,500 metabolites, 1 million DNA methylation markers. I will tell you right now, if you were to do all of those same measurements on one patient, it would probably cost you somewhere between $15,000 to $20,000, which is not feasible, which is why we are doing that work for you, and then being able to really give you those outcomes with, a very cheap kit cost, a quick turnaround time in just that one blood spot card. So I went over, the idea of this omic MH, how we created. So what makes this superior, as I was stating? Well, number one, omic MH is better at predicting outcomes. It really outperforms all previous DNA methylation clocks in predicting death and odds and hazard ratio to disease. All right. You can see here what we trained the omic MH with, this little pink dot, the DNA methylation EMR age. Is going to have a higher odds ratio, as I mentioned, and hazard ratio compared to all the other clocks, except this little one right here, COPD. That is going to be the grim age, the death predictor, which really used to be the gold standard because it was trained using smoking pack years. So it's going to be better at predicting COPD. You can also see in this image here that the omic MH is going to have a better 5-year survival prediction and 10-year survival prediction compared to any other clocks created. The omic MH can predict when you are going to die with a 92% accuracy rate compared to chronological age, which is only 72%. We do not report out on that. We absolutely could. We choose not to at this time, I think, for obvious reasons. But again, that is the point of these algorithms, is that they have to be predictive, so it initiates some type of change for you in the clinic. Secondarily, omic MH is extremely reproducible. This was an issue in previous clocks. Remember, we do not want to talk about accuracy when it comes to these clocks. When you're accurate, think of throwing darts. You're getting something. You are getting to bullseye, right? You're getting to something that is known. Biological aging is not known. So we want to be very, very precise, and we can make sure that these clocks are precise by using what we call an ICC value, or interclass correlation value, meaning if you take two samples at the same exact time, how highly correlated are they to one another? And in this case, this is a very blurry image, but the R-squared value is going to be 0.995. All right, pretty much perfect here, which is exactly what we want to see. These need to be reproducible. You need to be able to retest over time to make sure what you're tracking is true change with the underlying biology and not just noise from the algorithm. Number 3, it's connecting acceleration and risk. So I already gave you a little bit of a hint here, but you can see we can give you your relative risk as it relates to death, cancer, heart disease, stroke, type 2 diabetes, COPD, and depression. All right. This will get better and better. We'll be able to detect more diseases and relate this to outcomes as the information becomes available. But this is a relative risk. It's not absolute. It's not telling you when you're going to get disease. It's a risk as it relates to that overall omic age. So we can report this based on the assumptions generated in the biobank cohort and the data we were able to gather from the longitudinal analyses. Number 4, this is really important as well. These are all very important. But number 4, the omic age is going to be correlated to the clinical picture and the pace of aging, that 3rd generation clock that I mentioned. The 1st generation clocks that companies use are often very unreliable, as we know it, at capturing a true biological signal. And this is because, again, chronological methylation clocks are correlated to aging, but they're not linked to what we classically know about the disease. So we now see consistency among the pace of aging in our omic MH, and this makes it for a much more intuitive and more consistent picture to clinical phenotypes. So, we often show, why would the person with all of the classical aging risk factors be slower than aging than someone without? Number 5, the omic MH is going to control for immune cell subset variations. One of the reports that you would also receive when taking a complete kit with TruDiagnostic is this beautiful immune cell subset outcome. If any of you have done immune cell subset testing in your practice, it is a logistical nightmare. It's a large plasma sample. It has to be shipped on dry ice. It's very, very expensive to you, thus passing that cost off to the patient. So we actually created a way to measure these immune cell subsets and to control for them, because traditionally in other biological age clocks, the immune cell subset variation leads to high biological age variation. And this is explained within these individual outcomes. So some research, for example, has really shown that human naive CD8 T cells, which actually go down during aging, have an epigenetic age that is 15 to 20 years younger than effector memory CD8 T cells, isolated from the same individual. So if you're using algorithms that don't control for these, it can vary dramatically. So to make sure we're not seeing major changes due to changes in the immune system, we have to incorporate this within the algorithm. And then you also get it as a side report as well to see where your immune cell subsets lie. Number 6, omic MH explains why you are aging. So this is really where it comes down to the EBPs, those epigenetic biomarker proxies, where I told you within the algorithm, we measured all of those proteins, metabolites, and clinical lab variables. Well, we can actually report these out individually based on the epigenetics and methylation alone. So these are surrogate markers. They are not to replace your clinical lab testing if you do proteomic or metabolomic testing at this time. Will they eventually? Probably and absolutely, but they are just meant to tell you how these outcomes are related to that overall omic MH. So for example, one that I show here is this blood urea nitrogen. Lower levels of blood urea nitrogen are associated with improved omic M aging. So this person in particular has a blood urea nitrogen score that's in the 90th percentile. So it's 90% higher compared to people of the same calendar age and sex in the TruDiagnostic cohort. We need to take corrective action to try and lower this blood urea nitrogen as much as possible. So with this, you're going to be able to be given more direct recommendations and things to point you to drive that lowering of the omic MH, where previously with those first generation clocks, again, that aren't controlling for a lot of these outcomes, you would make just more very general epidemiological associations or recommendations. So now I want to show you how these are actually associated to disease, right? I need to show you that what we measured in that omic MH study is actually true. So this is a heat map. You may have seen some of these heat maps before, but these are showing associations of biomarkers measured in our omic MH study to outcomes. So EVP associations on the x-axis with the disease from the biobank cohort, or other associations, like degree, how many times you're working out in terms of exercise per week, hours of sleep, on the x-axis, the EBPs on the y-axis. And the effect size, meaning if something is red, that's a positive association. As one goes up, the other goes up. If it's blue, it's a negative association. As one goes up, the other goes down. So what do we see here? Well, here we see HbA1c and fasting glucose. Positive association here with type 2 diabetes. This makes sense, right? This looks awesome. Glucose, HbA1c is going to be positively correlated with type 2 diabetes. Another correlation we found is going to be with the insulin-like growth factor binding protein 2 and basically your weight status. So the insulin-like growth factor binding protein 2, it's in the family of the IGF-1s. And as this is increased, as it is overexpressed, people are going to be resistant to the development of obesity and insulin resistance and vice versa. So you're not expressing this as much. That is going to lead to obesity. All right. Finally, I am getting towards the end here. I'm going a little bit quicker than expected, but I want to show you just a couple other associations that we found as well, and hopefully ones that you may be a little bit more familiar with. This is a metabolite that the omic MH selected out of the 8,500 metabolites we measured, and this is carotene 3,3-diol or lutein. This is one of the 600 known naturally occurring carotenoids, and it is synthesized only by plants. It's found in really high quantities in green leafy veggies like your spinach, kale, yellow carrots. Some studies have even shown that this can improve cognitive function and eye health. So you probably hear people say all of the time, eat more carrots, right? It's going to help with your eyesight. It's because of this little metabolite right here, this carotenoid. There are, several papers, one in particular that's probably notable to mention here, that was a large meta-analysis involving about 71 published papers that represent more than about 400,000 individuals, showing that people who have higher lutein intake or higher blood concentrations of lutein, again, have a reduced risk of coronary heart disease, stroke, and metabolic syndrome as well. So those are some of the disease outcomes we can relate it back to. And classically, this has always really been related to aging, if you research it. On top of that, it's also been implicated in visual function. It's, protective against many age-related eye diseases, like age-related macular degeneration and cataracts. More recently, there's been insight into the bioavailability of lutein, which have led a bunch of researchers to consider a role for lutein even in like brain development and cognitive enhancement and performance. So this screenshot here and what this slide is really showing you is that it's a screenshot actually from Dr. Michael Lustgarten. If you're familiar with him, he has a channel called Conquer Aging or Die Trying, basically showing you that the carotenoid was a measurement used in GrimAge, or that second generation clock, that was created even a couple years ago. So it's showing you that, GrimAge is picking this up as being significant, as well as our omic MH. So we know a carotenoid, a high level of carotenoids or intake of these metabolites are going to be associated with a younger epigenetic age and reduced all-cause mortality risk. My last example here is one that I think you will really enjoy and find pretty interesting. Have any of you all seen the Netflix documentary series, You Are What You Eat, by chance? The twin study, possibly? It was like the vegans versus omnivores, identical twin study linking plant-based diets to better biological aging. If you have, TruDiagnostic actually was heavily involved, and you'll see some of our research presented there. But essentially what they did, it was a study that used a randomized clinical trial design involving 21 pairs of twins who are either put on an 8-week entirely plant-based or vegan diet and a healthy omnivore diet. And these samples were, collected from the participants at the start and the end to measure, DNA methylation. And you all probably want to know the results, which I'm happy to talk about, but that's not really what I'm highlighting here. I'm going to highlight individual epigenetic biomarker proxy changes that we saw. But if you do want to know the results, the vegan diet group actually showed significant decreases in overall epigenetic age acceleration in some of those really popular second generation clocks in the deneen pace, as well as systemic areas such as decrease in inflammation age, heart age, hormone age, liver age, and metabolic systems age. But I do want to, also let you all know that even though we saw those results, we still need more information. So it's really crucial that a vegan diet, lacks essential nutrients for optimal epigenetic processes, right? So without proper supplementation, there's always risk of long-term damage. So, really as scientists, it's our duty to communicate those messages very clearly, empathetically, to make sure, the public's well informed about the nuances of dietary choices for their overall health, and they're going through a healthcare provider to make that decision. But back to, what we actually found, it was super interesting. You can see that I populated these images and these studies here on the right-hand side, and it's actually looking at metabolites and proteins that the omic MH chose as being statistically significant and correlated with aging that you can see here, that are going to have a significant p-value. One in particular that we can talk about is this DASH diet, the dietary approaches to stop hypertension, decrease CXCL4 concentrations among patients with stable CAD. And this particular protein is also negatively correlated with vegetable intake. So again, we're finding things in studies that we're doing today that are already long documented from previous, again, peer-reviewed literature. So what we're starting to uncover is that epigenetics can really be used to report out other areas of medicine, other multi-omic layers. And that is that really is the key takeaway. In addition to that, to really end hopefully on a positive note, you all absolutely need to be able to measure this number one risk factor, aging, in order to manage it. Everything else should be secondary. We know that epigenetic DNA methylation is the best way to measure biological aging at this time. We don't see anything, I think, surpassing it unless a new technology is created. Keep in mind that epigenetic DNA methylation is more than just biological age. And that's really important because, again, that talks about the future and the excitement of where these markers are going. We really want to call it, we're not a biological age testing company. We're an epigenetic testing company, where epigenetics can do just much more than aging itself, even though you probably heard of us or became initially interested through measuring aging. And the number 4, omic MH, is the best way to understand how and why you're aging. So with that, I will leave it up to you all for questions. But if you're ever interested in, setting up a call with me or someone from my team, please feel free to reach out. That's my email there. I have a couple other platforms as well, and even have a podcast called Everything Epigenetics if you're more interested in learning about what these markers mean from a research standpoint. So I appreciate you all listening, and hopefully we can have a couple questions and more fruitful conversation later on.
Thank you very much, Hannah, for your presentation. I don't think anyone is questioning the hard work, and that this really is a solid test that gives a lot of information. But I think a lot of us sitting here, having heard what your presentation, is thinking, okay, so if I do the test, what am I actually informed about, and what can my action be? What's the clinical? One thing is getting some information, but what's the clinical or the tool that we get out of it in order to do health optimization in ourselves or in our patients?
Yeah, definitely. And I think that would really maybe be helpful by first showing you some of the reports, or maybe what those look like, if that's okay, and kind of what's included there. So I can definitely walk you all through those reports. With anyone who ends up taking a test, I would highly recommend setting up a one-on-one call with me afterwards to go through the results. We do have supporting, education, documentation on how to review these results and really dive into them. But it's always, better to I think do this one-on-one just to get you up and running with the testing. I think I probably first, though, want to mention even the 2 products, or really what you're going to get. And those 2 products, let me just put this image somewhere else here. The 2 products are going to look like this. Let me share my screen again. Oh, well, that doesn't do you any good if you can't see it. The TruAge Complete Kit and the TruAge Pace Kit. So the TruAge Complete Kit is going to include everything. You get your omic MH, that deneen pace, 12-cell immune deconvolution method, telomere length. Those are the main age outcomes. You do get some disease-based reports as well, and some characteristic and trait-based reports you can see here. Most people do this as a baseline first, and then they do the TruAge Pace as a follow-up with the deneen and the telomere length. You can do this every 2 to 3 months. You can do the complete every 6 to 12 if you really wanted to. And what you are going to get back, can you all still see my screen?
Yeah.
Yeah. Okay, perfect. What you all are going to get back is a lot of reports if you do that complete kit. And they are all very, very useful. They explain a lot of information on how you're going to go over these. But really, clinically speaking, you're probably going to use the summary report the most, the healthcare provider summary report, which is more of a quick hitter of all of the results. And then you're going to use that expanded omic MH report, which goes into those individual EBPs or biomarker proxies. So again, and Catherine, when you were talking about clinically, how are you going to use this? Most of our providers here in the States are going to include this as a baseline primary test for every single one of their patients initially, and then remeasuring again every 6 or every 12 months to see how the therapies you're doing are affecting that pure reading of age. And maybe it's up to the patient as well, right? It can speak a lot to how the patients are complying, if they're following your regimen or what you're actually recommending. Even from a lifestyle standpoint, we report out in this complete kit how much the patient has smoked and drank, consumed alcohol across their entire lifetime, right? So we're not shaming anyone and telling you don't drink or don't smoke. That's ultimately up to you and the patient. But we are saying, hey, we're able to detect this amount of smoking exposure, this amount of alcohol exposure, and just quantifying that for you. So it can really drive patient behaviors, I think, is a big point as well. It really puts it on paper, that biggest risk factor in a quantifiable form that's really tangible.
So, so in the report, you will get ideas so of course, when it's smoking and drinking, I guess it goes without saying that if there's a lot of damage, you can say, based on that, if you've been exposed to a lot, then of course it goes that we shouldn't drink and smoke, and none of us really should, depending on, doesn't really matter where that is on the if we want a long and healthy life. So that's a little bit, it kind of goes without saying. But some of the others in relation to exercise and diet, etc., can you just maybe come with some examples there?
Yeah, absolutely. So for example, let me pull this up here. Right here, the omic MH report, right? So this is where you would get that omic MH, which is going to be the most predictive algorithm. You can see, some images from the presentation, some background where you would get that age. And then you can scroll down and look at those individual EBPs or those biomarkers, right? And see which things are out of range. For example, if we see maybe red blood cell distribution with being too high, hematocrit and hemoglobin being too low, there are definitely some deficiencies that we would point out there from a nutritional standpoint. Just to go on that example, and Catherine, there are plenty more to give, where we may say you're definitely deficient in things like iron, right? Vitamin B12, folate, we may recommend supplementation there. Even things like vitamin C to enhance iron absorption, citrus fruits, tomatoes, peppers, etc. We're currently working on EBPs for a nutritional panel. So again, you can really report out anything through the lens of DNA methylation and epigenetics. We probably have over, 100 new business ideas here every single day. But one of them that we're working on is actually reporting out basically think more of like nutrigenomics, right? But through the lens of epigenetics. Another kind of outcome in particular, and Catherine, if you want to, harp a little bit more on like dietary recommendations and relating this to outcomes, this red blood cell distribution width is more of like an anti-inflammatory marker, we believe, really measuring inflammatory status and burden. So, you may want to adopt an anti-inflammatory based diet, right? Engaging in regular exercise, as you mentioned, adequate sleep, right, to help manage that chronic inflammation too.
So that, some of the questions are coming up now. They weren't, so I have to read them as they.
No worries. I can read through as well and see if there is.
So.
I think maybe just I just saw one pop up from Leah saying, "Are there any training programs for practitioners?" And Catherine, I can send you all of our webinars and seminars in case you want to send those to the providers. That way they can go through and listen again or learn a little bit more. In terms of qualifications or certifications, there really isn't anything yet, but we are working on creating one. So it's so new that the education behind these are very limited, but we will work on getting that up and running for you. It's something that we've been wanting to do for quite a long time. So stay tuned, but that should probably be available, even within the next 2 to 3 months to become certified.
Super. So just there was a question in regards to how does this differ from DNA health? So we have the DNA test that's looking at SNPs and it's looking at the genes and not going to change, where this is the epigenetics and the expression or like the health status in real life. So it's a really good test pair. Both the DNA health will of course give you clues as what areas that are genetically not possibly not functioning optimally. So it's not a direct comparison, but it's both information that can be relevant to have in order to understand yourself most possible.
Yeah. I think adding to that too, Anne Catherine, this is one of the images I wish I would have had on my presentation that I thought I did. But going back to just the definition of epigenetics, epi is a Greek prefix. It means above. So think of us looking on top of the DNA. We're not actually measuring any genetic material. We're not actually measuring SNPs. Most people relate methylation to MTHFR SNP, right, into the COMT SNP, but those are going to be genetic variants. And it's still very important that you're measuring your genetic makeup. I recommend using even these two tests in tandem as a baseline. It makes for a really nice, well-rounded package. But in particular, there are a lot of epigenetic modifications that can happen. TruDiagnostic is focused on measuring the DNA methylation, meaning we're measuring just a CH3 group, right? A carbon and 3 hydrogens. And that's going to be located on your on what's called a CPG, or a cytosine guanine nucleotide combination with a phosphate bond holding them together. And by definition, if something is unmethylated, the methylation is not there. We always go back to kind of this light bulb example. The light switch is on. The light is shining. Your gene expression is turned on. Whereas if something is methylated, the light is turned off, no light, your gene expression is turned off. So it's not necessarily saying methylation is good or methylation is bad. It is very site specific. It depends on the marker we're looking at, where, we want our oncogenes to be methylated and turned off, but we want our tumor suppressor genes to be unmethylated and turned on, right? So it's all about, combining these different age-related outcomes and a synergistic approach to make recommendations that are going to, again, shift those methylation markers in your favor.
Exactly. So it's the thing that like genes, it's the interpretation of the gene, and of course here it's the interpretation of the activation of the gene and what goes on biologically in the body. So this is clues to understand the body more, clues to, because someone asked, well, I mean, do you really need kind of, I think now I'm maybe interpreting the question a little bit, but I mean, we're talking about diabetes, cardiovascular disease, we're talking about aging. Do we really need a test to know this? And of course, no, we can just live healthy and do the exercise that we have to, etc., and do the best we can without this information. But if we want to do biohacking and we want to do health optimization, then we want to, of course, understand and test our genes. And we, of course, want to do these biological age tests so that we get an idea, where can we hack, where can we improve, what else should we, are there any specific supplements that we can add to the pack, stuff like that. So I guess for the person who is less hungry for knowledge, then it doesn't really matter to do a test like this. But for everyone here who's been now sitting here speaking or listening to Hannah's amazing speed talk presentation, I mean, then it matters. So it's a little bit of question like, how much information do you need to know? Are you happy with a used secondhand car, or do you want to just get the newest or the most, I don't know, out there? So it's all a question of interest, and of course you have patients that won't even benefit from this because it's just a question of like just getting enough vegetables on the plate, or just moving enough, and then we don't, I mean, this will just be talking above their heads, and it probably won't motivate them. But then there's others who just need this information because it will motivate them. So again, as the clinician or the provider or the practitioner, you need to choose when to use what tools. So I have patients where this is just, whoa, wow, information, amazing information, let's work on that. And I have others where doing a vitamin D test is more or less overwhelming them. So we as professionals needs to evaluate what patients can we use these tests and this information for. And perhaps you would be the first, of course, guinea pig to try it out on yourself. I know I did, and I was overwhelmed by the information, but I thought it was an amazing tool to carry on working with. So that's probably my take on it. But reach out as you get the testing done. If you need any support, then of course we can, I mean, because Nordic and DNA Live won't be able to educate the way as Hannah's team does. So we'll find a bridge or a collaboration so that, and exactly the education material, we'll share it, etc. So, but there is always new tests coming on, there's always new research coming in, and I believe that everyone on this call is always hungry for new information, new knowledge in regards to expanding longevity and have the longest and healthiest life as possible. Thank you very much for your time, Hannah, and everyone out there. It's always lovely to have this Thursday for me afternoon with you. It's a special hour for me where we're so many like-minded together. We will share the recordings, slides, links, what have you, as much as we can. Hannah, thank you very much for a sterling presentation. We will be in touch.
Yeah, awesome. Thank you, everyone. I hope to hear from some of you soon. My inbox is always open, so it's wonderful to meet you all. Thanks so much for setting this up and coordinating, Anne Catherine.
Thank you. Take care, everybody out there.
Bye.
Bye. Bye.
If you do not already have an account with us, please contact our team and we will assist you. If you found this webinar helpful, please like and subscribe to our channel.